Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Balkis. My metric number 23DVT21F1039. Today, I want to do the presentation about glycolysis. Glyco, more to glucose. This is breakdown. Occurs in the cytoplasm of the cells. Molecules of glucose are broken down into two molecules of pyruvic acid. Cell must use inverse 2 ATP. Produce energy carrier molecule. First 4 ATP, third 2 NADH. Splitting of sugar is which CG glucose is split into 2 3C sugar. The smaller sugar are then oxidized and their atoms are reached to form two molecules of pyruvate. She also do in an aerobic catabolism of glucose. The first step is cellular respiration, take place in a cytosol. The pathway of glycolysis is divided into two space phases. First phase is energy investment phase. There are required energy endogenic reaction in the form of ATP. Two molecules of ATP are used in this phase. Energy payoff phase. Energy yielding phase. The agonic reaction 4 ATP and 2 NADH are produced. Thus, the net energy yet from the glycolysis in 2 ATP and 2 NADH per glucose molecule. In this picture, what we see is the step of glucose made to produce the pyruvate in a flow chart. The first step is phosphorylation. The ATP depends phosphorylation of glucose to form a glucose 6-phosphate categorized by hexokinesis. Second step is isomerization. The glucose 6-phosphate is reagent to become a isomer fructose 6-phosphate. Step 3 is phosphorylation. A second ATP is used distillation to phosphate glucose 6-phosphate to form glucose 1,6-biphosphate. A step is categorized by phosphate fructose kinase. Step 4 cleavage. Fructose 1,6-biphosphate is hydrolyzed to the form 2,3G. Step 5 is isomerization. Succeeding reaction of glycolysis utilize G3P as a substance. Thus, DHAP is isomer to form 3GP. The two molecules of 3GP then undergo identical reaction in the energy payoff phase. Step 6 oxidation, hydrogenation, and phosphorylation. Each C3GP molecule is oxidized and NADH plus is reduced to form NADH plus H plus. The product phosphorylate reaction is inorganic phase to the yet a higher energy substance 1,3-biphosphorylate. Step 7 substrate level phosphorylation. Once phosphorylation group from each 1 phosphorylate molecule is transferred to the ADP to form ATP and 3-phosphorylate. Transfer a phosphorate group from a phosphorate in the intermediate are known as certain level of phosphorylation. Step 8 is isomerization. 3-phosphocrate is ready to form 2-phosphocrate by shifting the position of the phosphate group. Step 9. Hydration. A molecule of water is removed to 2-phosphocrate to form another high energy compounds. PEP. PEP has phosphate group attached by an unsubstantial bond. The last step, step 10, is a certain level phosphate. The phosphate group from the PEP is transferred to ADP to form ATP. PEP is the cover to private.
Okay, good afternoon all. Today I will talk about Krebs cycle. So now we will see their steps. Okay, so in the Krebs cycle there are eight steps. So I will explain one okay. So step 1 is condensation. So in the step 1, the cycle will start with the breaking of the unstable bond attaching of the acetyl group to coenzyme A. The acetyl group then combines with a 4 carbon molecule oxidated to form 6 carbon compound. As an example, citric acid or citrate. Okay, now we will talk about step 2. Atoms of a citrate are rearranged by the removal and addition of water molecule. As a trait, the isomer of citrate is formed. Okay, the step 3 is citrate is oxidized and loss of molecule of carbon dioxide, forming a 5 compound A ketoglutarate. So NAD plus is reduced to NADH plus H plus. So now we will see the step 4. The step 4, A ketoglutarate is oxidized and loss a molecule of carbon dioxide and combines with coenzyme A to form a 4 carbon compound. As an example, succinyl CoA. Okay, in the step 5, the bonding attaching coenzyme A to succinyl CoA is unstable. So the bond breaks and coenzyme A is displayed by a phosphate group, which is then transferred to guanosine phosphate GDP to guanosine 3-phosphate GTP, a compound which is similar to ATP. GTP then transferred its phosphate group to ADP, yielding ATP. In this reaction, succinate is formed. Step 6, succinate is oxidized and FAD is reduced to FADH2. The resulting compound is fumarate. Step 7, fumarate is converted to malate by the addition of water H2O. So in the step 8, lastly the malate is oxidized to reform oxalate, while NAD plus is reduced to NaOH plus H plus. So the regenerate oxalate can now combine with another molecule of acetyl coenzyme A, and the cycle is repeat. Okay, so that's only from me. So thank you.
Okay. Electron transport chain. In the electron transport chain, electrons are passed from one molecule to another, and energy released in this electron transfer is used to form an electro electrochemical gradient. The electron transport chain is a cluster of proteins that transfer electrons through a membrane within mitochondria to form a gradient of protons that drives the creation of adenosine triphosphate, ATP. ATP is used by the cell as the energy for metabolic processes for cellular function. The electron transport chain is used to pump protons into the intermembrane space. This establishes a proton gradient, allowing protons to be pumped through the ATP synthase in order to create ATP. This method of ATP production is called oxidative phosphorylation. The electron transport chain activity takes place in the inner membrane and the space between the inner and outer membrane, called the intermembrane space. The three main steps in the electron transport chain are: one, first generation of a proton gradient across the mitochondrial membrane. Proton accumulation occurs in the intermembrane space of mitochondria. Second, reduction of molecular oxygen and formation of water. And third, ATP synthesis by chemiosmosis. Why electron transport system is called oxidative phosphorylation? In the mitochondrion, what the proton gradient does is facilitate the production of ATP from ADP and PI. This process is known as oxidative phosphorylation because the phosphorylation of ADP to ATP is dependent on the oxidative reaction occurring in the mitochondria. Why is oxygen needed in electron transport chain? Oxygen is the final electron acceptor in the electron transport chain, which allows for oxidative phosphorylation. Without oxygen, the electrons will be back up, eventually causing the electron transport chain to halt. Oxidative phosphorylation is a made-up of two closely connected components, the electron transport chain and chemiosmosis. Where does electron transport and oxidative phosphorylation occur? The mitochondria. Oxidative phosphorylation occurs in the mitochondria of all animal and plant tissues and is a coupled process between the oxidation of substrates and production of ATP. As the Krebs cycle runs, hydrogen ions or electrons are carried by the two carrier molecules NAD or FAD to the electron transport pumps. Oxidative phosphorylation is the process by which ATP synthesis is coupled to the movement of electrons through the mitochondrial electron. the mitochondrial electron transport chain and the associated consumption of oxygen. The major step of oxidative phosphorylation is mitochondria include first, delivery of electrons by NADH and FADH2. Reduce NADH and FADH2 transfer their electrons to molecules near the beginning of the transport chain. Second, electron transport and proton pumping. Third, splitting of oxygen to form water. And last one is ATP synthesis. What is oxidative phosphorylation and why it is important? Oxidative phosphorylation provides most of the ATP that higher animals and plants use to support life and is responsible for setting and maintaining metabolic homeostasis. Why is oxygen important in oxidative phosphorylation? Because in oxidative phosphorylation, oxygen must be present to receive electrons from the protein complex. This allows for more electrons and high energy molecules to be passed along and maintains the hydrogen pumping that produces ATP. The last one is what is mitochondrial oxidative phosphorylation. Oxidative phosphorylation OXPHOS is the main source of energy in eukaryotic cells. This process is performed by means of electron flow between four enzymes, of which three are protons pumped in the inner mitochondrial membrane. That's it. Thank you.